We are getting ready for the EOC. This is your chance to really practice to get ready to perform big on that end of course test. I know that if you study, if you complete this study packet and study, that you will do great on the EOC. So when we look at this section, we do want to write, here it says objective 4.1, compare the structure and function of organic molecules in organisms. And so what is the structure, what is the shape, and what is the function, what is the job? In our handbook, this was section 1.1. Now I'm not going to tell you the functions. The functions are actually in your notes. I am going to tell you the subunits. Remember PCLN, AA, MTN. Please come late, Nate, and ask me to narrate. Now we wrote the sentence in a different order, but if you remember it in this order, you'll remember that carbohydrates are made of monosaccharides, and that proteins are made of amino acids. Lipids are made of triglycerides, and nucleic acids are made of nucleotides. And so as you go back, you want to look at what is their job. For example, carbohydrates' main job is to be a source of energy. But you'll find all of these jobs in your notes for section 1.1. The next section looks at specific examples. I do want you to write carb here. Starch and cellulose are both carbs. Insulin is going to be a protein. Glycogen is going to be a carb. Glucose is a carb. Enzymes are a protein, and hemoglobin is a protein. And so, if it is a carb, that means that the subunit is a monosaccharide. Now, starch is actually used to store carbs in plants.
Kadeja Elleen, please come to the front office. Kadeja Elleen, please come to the front office.
So carbs are used to store, starch is an example of a carbohydrate, and it's used to store the carbs in plants. It's like storing their food for later. Cellulose is used to build the cell wall of plants. And since it's a carb, we know what its subunit is. Insulin is a hormone to control blood sugar. I'm filming a video, I'm sorry, I can't help you right now. If you look at glycogen, glycogen is a carb, but it's used to store carbs in animals. For glucose, enzymes, hemoglobin, and fats, you're going to go back to your notes, section 1.1, to see what you can find in your notes. Remember, you took a lot of time this year to take your notes, so it's important that you're using them and studying them. You also have RNA and DNA, and again, it's directly in your notes in section 1.1. As we move down, we look at nutrient tests. Where it says nutrient tests, I'd also like for you to write indicator. These are our 4 indicator tests. So for starch, we use something called iodine. And iodine, when it's just in water, so let's write this here, in water, that would mean there is no starch if iodine is just in water. When it's just in water, it's really a yellow. But if it is in starch, if we put iodine in starch, it turns blue, black. And And so as you go through starch, lipids, monosaccharides, and proteins, each of them had a test. Now we do want to be careful with monosaccharides. That's things like glucose. And that's the one where we use Benedict's. But we also had to add heat. So you do want to go back and look at each of these indicator tests. Now for some of you, you might want to pause the video now and look it up in your notes. Others of you might want to let the video keep playing and then go back and find it. That's up to you. Objective 4.1.3, develop a cause and effect for the specificity of enzymes. Specificity means that it's specific, that they only work with certain substrates. So, explain why enzymes are specific. We want to remind ourselves that their active site is shaped for one substrate. Now here's where we have to remember this word, substrate. If you look down here, A, B, C, and D, where it says molecules, please write substrate. And next to substrate, write start. These are what we start the reaction with. Remember that an enzyme speeds up a chemical reaction. And so when we have a chemical reaction happening, the enzyme helps it happen faster. But each enzyme only works with one substrate. So if we look here, this is the only substrate that will fit into that active site. And that's why enzymes are specific. When it says explain the importance of the shape, I want you to answer what will happen if we lose the shape. What will happen if we lose the shape? And so this, I need you to answer. What happens if the shape changes? Now this shape is determined by the protein. And so because enzymes are protein, the shape is determined by the proteins or the amino acids that we put in order. So if you remember from one of the other units, when we put amino acids in order, they form a protein that is a specific shape. And then you've already answered which substrate would fit. And that's the end of page 1. What do enzymes do to the rate of chemical reactions? They are going to speed it up by lowering the activation energy. So we have all of these molecules in our body, proteins, carbs, lipids, nucleic acids, and they each have a job. And one of the really important jobs of proteins is to be enzymes. Some proteins are enzymes that speed up reactions. If you look here at the word rate, rate means speed. So how quickly is this enzyme happening? This enzyme is taking place best right here. And optimum means best. So the best temperature would be right about here. So you do need to answer that. And again, we want to look at the best pH. And so you should be able to fill that out. Remember, if I change the temperature or I change the pH, I denature the enzyme. So while enzymes are supposed to be doing all these things in our body, like breaking down food, if the temperature isn't optimum or best, then the enzyme can't work at all. And so that's the question here. How do extreme pH and extreme temperature affect enzymes? They can change the shape of the active site, and we call that being denatured. It does ask you next in the notes, to look at the common characteristics of enzymes. And so this last section is really going to look at enzymes. And so you want to go back and fill these in. And that's actually the end of this first section of notes. And so you want to stop the video and make sure that you've gone back and filled out all those parts.